In today's class, we'll see what is pharmacology actually. So, pharmacology is a branch of science that deals with what a drug does to the body and what a body does to the drug. So, we are taking many medications, we are taking many drugs into our body. So, after going into the body, what is happening to the drug? What is our body doing to the drug, and what is the drug doing to our body? Let us see what is drug first. So drug is any substance or chemical compound once after administered which show its biological properties by altering the pathological and physiological properties of the body. So here a drug, once if we take any drug, if the biological system, whether it may be pathological or physiological, alterations are taking place in the body, and if there is any biological functioning is altered, so then that kind of the chemical compound is called as a drug. So WHO gave a definition as drug is any substance or product used to intend to be drug is any substance or product used or intended to be used to modify or explore the physiological state or pathological condition for the well-being of the recipient. So here what? So the physiological or pathological changes for the well-being in order for a person to be cured or to get out of any discomfort which is occurring in the body. So that compound or chemical substance is called as a drug. Here is the pharmacology. What does a pharmacology deals with? So it is a branch of science which is involving the pharmacokinetics, pharmacodynamics, pharmacotherapeutics and pharmacognosy. So pharmacokinetics in the sense what the body does to the drug. So here once the drug is entering into the body, it undergoes various processes called as absorption. Once after it's entering into the body, it gets absorbed. Then after it is getting absorbed, it gets distributed all over the body. And after it is getting distributed, whatever the remaining or the leftover product is there, it is getting metabolized. So the metabolic products, after metabolizing, the metabolic products whatever are formed, they are eliminated out of the body. So this is an ADME process, that is absorption, distribution, metabolism and elimination process. So this is what our body is doing to the drug. So if we see pharmacodynamics, what is the drug doing to our body? So once we take drug, any drug is having side effects. So at least the common side effects like headache, dizziness or drowsiness or sedation or whatever it may be, these are the common side effects of any drug. So here after entering into the body, the drug, because of some chemical reactions which are going into the body, it may cause some interactions. So here the drug, if we are taking 2-3 medications, the drug may interact with the drug, or else if we are taking the drug along with the food, the drug may interact with the food. So here there are various things a drug is doing to the body. And the other one is pharmacotherapeutics. It is the study of use of drugs. So how the drugs are used or what are the clinical uses. So each and every drug is not used for every disease or every discomfort. So here each and every drug has its own clinical application or clinical use. So pharmacotherapeutics is nothing but how far the drug is useful in any disease. And the other one is pharmacognosy. Here the pharmacognosy is considered to be a branch of pharmacology because there are the naturally occurring plant compounds which are useful as the drug. So here we are identifying the therapeutic compound which is essential as a useful component in order to treat a disease. So that is the reason pharmacognosy is also a branch of pharmacology. And the other one is toxicology. So toxicology is a branch of science which is dealing with how far the drug is toxic to the human being. So what is the dose or how much dose we can take in order for the drug to be non-toxic. So these all are involved in the branch of pharmacology. So let us see the sources of drugs, from where these drugs are obtained. So there are many sources of available, there are many sources for a drug to be extracted. So a few of the sources are plants. From plants few drugs are obtained, from animals, from minerals, even from microbes, that is microorganisms, synthetically compounds are prepared, and through genetic engineering technology. If we see the sources, a few of the examples of the drugs here, from plant source, from the plants like berries, I mean from the seeds or barks or leaves of the plant, or even from resins. Resins are the gum substances which are obtained from the plant, and even from the roots. So from all these parts, each and every part of the plant is useful in order to extract the drug. So for example, if we take morphine, morphine is obtained from quinine is obtained from bark, and digitalis from leaves, and gum acacia from resins. And the other one is minerals. So these minerals are richly available in earth and as well as soil. So from the soil we are extracting the minerals, or from the groundwater we are extracting the minerals. So here the minerals like iron, sulfur, potassium, silver, and even gold are extracted for clinical use. And from animals, so from animals, from the glands of the animals or from the organs of the animals, we are extracting the crude materials, or furtherly they are processed as a drug, which are like pork insulin. Though the human insulin is obtained from the pork. So here the synthetic compounds are which are man-made. We are synthetically preparing few of the drugs from the analogs. So here if we take for example morphine. So morphine is a naturally occurring plant compound. We are synthesizing morphine analogs for the better effect and better potency than morphine. So the examples include meperidine, sulfonamides. The other source is that microbes. So even from fungus, from virus, and even from bacteria, we are extracting the important compounds. For example, if we take the fungus substances and streptomyces and even bacteria. So the antibiotics like penicillin, erythromycin, as well as bacitracin. So these are all which are obtained from the microorganism source. Now let us see from where, or else we can say what sort of compounds are obtained from the plant. So if we see alkaloids, so alkaloids are the naturally occurring plant compounds which contain the nitrogen based structure in their nitrogen based ring in their structure. So if we see the structure, they contain the nitrogen group in them. That is the reason they are termed. Usually if we see, in order to identify any alkaloid, at the end they are ended as Ne. For example, if we take morphine, morphine contains alkaloid. Quinine, it contains an alkaloid. So these are ending with Ne. So such sort of compounds or such sort of drugs are containing alkaloids in them. So this is one way to identify what sort of plant source is present in them. The other one is glycosides. So glycosides, they contain the sugar moiety, or else they contain the glucose or fructose or lactose in their structure. So they contain a sugar moiety. Along with that, a non-sugar moiety is there. So one will be a carbohydrate, the other one may be a fatty acid or amine or ester. So they contain a non-sugar moiety along with the sugar moiety, and these are connected with the ether linkage, that is C double bond OO, COO linkage. For example, if we take digoxin, so digoxin is a compound, or it contains a glycoside which is obtained from the plant called as digitalis purpurea. The other one is gums. Gums are, we can say, these are the waste materials of plants which are useful for us. So these are the waste materials. So in plants also there occurs various metabolic reactions. So because of the metabolic reactions in the plants, they remove, or how does a body eliminate the waste products from our body? At the same time, plant also eliminates the waste products out of this body. So whatever they are forming like a gum or sticky substance. It is an exudate of a plant. So those are called as gums, and these are generally used as emulsifying agents. So in if we see a formulated preparation called as emulsion, it contains two immiscible phases, an oil phase and a water phase. So these two are not miscible, they are not mixed up with each other. So that is the reason we have to use some emulsifying agents in order to mix these two immiscible liquids to form an emulsion. So if we take gum acacia, example is that gum acacia. So from this acacia plant, from the gum of the acacia plant, this gum acacia is extracted, which is a amorphous powder. So it is a smooth powdery substance. The other one is tannins. So tannins are the nitrogenous derivatives. So they contain nitrogen in their ring, but they do not nitrogen in the structure, but they do not contain the phenolic ring. So if we see alkaloids, they contain phenolic ring with the nitrogen group, but here they do not contain phenolic ring. So that is the reason these are non-phenolic nitrogenous derivatives, and these are used as astringents. If we take the example tinge and catechu. So catechu and tinge, these are the type of tannins which are extracted from the plants. And the other one is resins. So resins are nothing but these are the polymers of volatile oils. So if the volatile oils, if we polymerize, in the sense if we add many polymers to that volatile oils, so whatever the polymers we get out of these volatile oils are these resins. If we take example benzoin. We see the plant sources. Here we can see purple foxglove, which is also called as digitalis purpurea. So this is digitalis purpurea plant, which is commonly called as purple foxglove. Here the leaves of the digitalis plant are used in order to extract the glycoside useful product. So here the glycoside is extracted from the leaves of this plant, and that glycoside is called as digitalis. And this drug, digitalis or digoxin, is very useful. The digoxin drug is very useful in order to treat congestive heart failure. We see the morphine, which is obtained from the poppy seeds. This is obtained from the seeds from the plant called as Papaver somniferum. So from this seeds of this plant, this morphine is obtained, which is used as an analgesic agent. So here this morphine is used as an analgesic agent which is obtained from this plant. Here we can see the pictorial representation of the plant. The other one is Artemisia. So there are various species of the Artemisia. So here if we see Artemisia annua. So this is commonly called as wormwood, and this plant is mostly obtained in China. So this is mostly available in China, and this is used to treat various, we can say this is used to treat various microorganism diseases, like if we take malaria. So in order to treat malaria, this drug or is the source or the drug which is obtained from this plant is used. So it is used as an anti-malarial drug, and there is a modern drug which is available called as quingosu. So this is a drug which is modernly introduced, or which is modernly discovered in order to treat malaria, and this is obtained from the plant called as Artemisia annua. Here we can see cinchona. Quinine is an alkaloid which is obtained from the cinchona plant. Here the leaves as well as the bark of the cinchona plant are used to extract this quinine. If we see atropine, the other drug is atropine, which is used as a sympatholytic drug, and this is obtained from the plant called as Atropa belladonna. The other one is cocaine. Cocaine is also used as an analgesic agent, and this is obtained from the plant called as Erythroxylon coca. If we see the tabular column, cinchona is obtained from the bark, and the drug which is cinchona's bark is used in order to extract a drug called as quinine, which is used as an anti-malarial agent. And from purpurea foxglove, which is also called as digitalis purpurea, digoxin is obtained. That is, digitalis or digoxin is obtained from the leaves of this plant in order to treat congestive heart failure. And the other one is morphine, which is obtained from the poppy seeds of Papaver somniferum in order to treat, which is used as an opioid analgesic agent. Even codeine is also extracted from the same plant. And the other one is Artemisia annua, which is used as an anti-malarial agent. And the other one is Atropa belladonna, from which atropine is derived from the leaves of the Atropa belladonna, and which is used as a sympatholytic agent. So now we'll see the animal sources. So what are the animal sources, or what are the drugs which are extracted from the animal sources? If we see cow, from the pancreas of the cow we extract insulin. So the insulin is obtained from the pancreas of the cow in order to treat anti-diabetes, or else in order to treat diabetics, in order to treat diabetes. And if we see from the sperms of the fish, there is a drug called as protamine sulfate, which is used as an antidote for heparin. So what are the side effects or the complications which are caused because of the heparin? That can be we can use this drug as an antidote for the heparin poisoning. The other one is from the intestine of the pig. From the pig intestine, the heparin is extracted, which is used as an anticoagulant. In order to prevent coagulation, then this heparin is extracted from this pig intestine, and even from the ox lungs. From the lungs of the ox, the heparin is extracted and which is used as an anticoagulant. If we see synthetic drugs, so we are chemically preparing those drugs, we are manufacturing the drugs chemically. So such drugs are pethidine and cotrimazole. Let us see the mineral sources of the drugs, or what are the drugs which are obtained from the mineral sources. If we see magnesia, from magnesium, milk of magnesia is obtained, which is used as an antacid. So in order to antagonize the acidity which is produced in the body, milk of magnesia is used, and it is used even for constipation also. The other one is iron. Iron is obtained, the drug which is obtained from iron is ferrous sulfate, which is used to treat anemia. So anemia is a condition where there is improper functioning of RBC or improper synthesis of RBC in the body. The other one is zinc, from which the zinc oxide ointment is prepared in order to, which is used along with the calamine lotion or in order to treat the skin related disorders. The other one is calcium, from which the calcium sulfate is obtained, which is used in osteoporosis. So osteoporosis is a condition where there will be bone deformation or where the bones become brittle and easily breakable. The other one is gold, from which the gold salts are obtained, which is used to treat rheumatoid arthritis. Let us see the microbial source. So what are the drugs which are obtained from the microorganisms? So if we see from fungi, we are obtaining the drug called as Penicillium notatum, and the drug which is obtained from the fungi called as Penicillium notatum, the drug called penicillin is obtained, and from actinomycetes type of microbes, the microbes like Streptomyces species, either it may be Streptomyces venezuela or Streptomyces aurifacius. From this, the chloramphenicol and tetracycline antibiotics are obtained, and from bacteria, especially from Bacillus subtilis, we are obtaining an antibiotic called as bacitracin. Now let us see the drugs which are obtained genetically. So if we see genetic engineering. So genetic engineering is a technology where we are mixing two sort of genes. So we are taking one gene from one source and we are taking another gene from another source. For example, we can mix human gene with a microbe gene. So this is called as recombinant DNA technology. So we are mixing two genes where the hybrid gene or genetic gene is obtained in order to produce better effects. So all the microbes don't have the same effect. Some may have anti-malarial action, some may have antibiotic action. So if we mix these two genes, then the compound which is obtained, it may have the both factors of anti-malarial as well as antibiotics. So this is just an example for understanding of the same. So here we are obtaining the hepatitis B vaccine. So here whatever the vaccine which is used to prevent hepatitis, that is obtained by a recombinase technology. The other one is human erythropoietin. So erythropoietin is a compound which is used to synthesize RBC. So here this human erythropoietin is obtained through the genetic DNA technology. The other one is human insulin. Even the human insulin is also obtained from the if we take pork, cow, if we take cow pancreas. So here through the recombinant technology only or genetic engineering technology only, this human insulin is prepared. The other one is abscessamab. So this is also one of the antiviral drug which is prepared through the DNA or recombinant genetic engineering technology.